I think we can start.
Good evening, colleagues. Thank you very much for making time to attend this webinar today. We do know that there are different commitments and lots of things vying for your time, but thank you so much. We appreciate you joining us today. Firstly, let me introduce myself. I am Dr. Ausi Nghikodana. I'm a GP based in Pretoria in Gauteng, and I have a passion for cervical cancer prevention. I am here today in my capacity as a consultant working with FIND and will be moderating this webinar. This is the third of a series of webinars that we started hosting last month, and we have 4 more to go. So do stay tuned for the next 4 that are coming. Before we go into details, let me clear some housekeeping issues so that we can flow smoothly. Firstly, only the speaker and the speakers and the moderator mics will be active. So we have muted your mics, but if there is a need towards the end, when we go to the question and answer section, we will be able to unmute. So for now, you will be muted. As we proceed with the presentations, please post your questions in the Q&A section. They will be answered at the end of the presentations. Also, colleagues, please remember to complete the evaluation form before logging off. We will launch the evaluation form before we conclude the webinar. Also, remember that all registered participants will receive a link to the recording of this session early next week. And also just a reminder that the session is accredited for 2 CPD points, and we will ensure that we directly submit the CPD for CPD points to the Health Professions Council of South Africa. So around about now, colleagues, we are probably some people might be wondering who is FIND. So just a little bit about FIND. FIND is a global, the acronym stands for the Foundation for Innovative New Diagnostics. It is a global nonprofit which is focused on advancing diagnostic innovation and improving access to affordable, reliable diagnostic services, especially for women and girls in low and middle income countries at the primary care level and within communities. Colleagues, today we are having, as I said, the third in our series of webinars, and the reason we are focusing on we've decided to launch these webinars is to ensure that we capacitate healthcare professionals in preventing cervical cancer, which, as you know, is one of the most preventable cancers you can find. So, colleagues, today, without further ado, let me get into the details of today. We are joined by 2 speakers in the webinar, Drs. Leslie Bramford and Dr. Atisha Maharaj. First up, we are going to hear from Dr. Leslie Bramford, who is a pediatrician and also holds postgraduate degrees in the social sciences and in public health. She has worked in child, youth, and school health in the National Department of Health since 2006, and she has extensive experience in knowledge management, policy, and guideline development. In addition, she has extensive knowledge in program design, implementation, and monitoring and evaluation of national health programs. She currently oversees the department's maternal, women, and child health programs, and also holds an appointment with the Department of Pediatrics and Child Health at the University of Pretoria. Dr. Bramford's work focuses on ensuring that women, mothers, and children survive and thrive through strengthening the delivery of women's maternal, newborn, and child health services at community level, primary health care, and hospital levels. During the COVID pandemic, she also played a key role with regards to COVID case management and in the COVID-19 vaccine rollout. Dr. Bramford is here today to give us an update of the mother-daughter campaign. Without further ado, I would like to introduce Dr. Bramford to you and also to welcome all of you and Dr. Bramford's team members who are amongst us. And at this point, I'll hand over to you, Dr. Bramford, over.
Thank you very much, and good evening to everyone on the webinar. If I can just confirm that you can hear me and that you can see my slides.
We can hear you, Doc, and we can see the slides. We wouldn't mind seeing you for a brief second before you start.
So I think my video has been disabled by the host.
Okay.
So
all
let
right.
me present, and then maybe if you can sort that out, I'm happy to appear.
Awesome. Thank you, Dr. Bramford. Go ahead.
So good evening again to everyone, and thank you very much for this opportunity. I have about 15 minutes this evening, and what I will be talking about is an update from the National Department. What are our activities that are being implemented as the Mother and Daughter campaign, which is really focused on elimination of cervical cancer. And predictably now my slides are not moving. So let me just see if I share again, whether they will start moving. There we go. So as I'm sure everyone on this webinar is aware, WHO, the World Health Organization, has 3 key targets for eliminating cervical cancer. And South Africa as a country has adopted these targets. And we know that there is an enormous amount of work that needs to be done if we are to reach these 3 targets. The targets being ensuring that 90% of girls are vaccinated against HPV by 15 years of age, that 70% of women are screened for cervical cancer, and that 90% of women who are identified with cervical disease receive treatment and care. So last year, the National Department of Health launched the Mother and Daughter campaign, which essentially has 4 objectives, which you can see are very closely linked to the 3 WHO targets. The first being to strengthen and expand the HPV vaccination program in order to reach that target of 90% of girls. Our target is to introduce HPV screening, so to transition from liquid-based cytology screening to HPV screening with the aim of reaching 70% of eligible women. So 2 parts to that objective. First of all, the transition to HPV screening, and then reaching more women with screening. Thirdly, improving treatment and linkage to care, trying to reach that 90% target. And then the 4th objective is to develop an electronic data management system to record individual level vaccination data to track and monitor the impact of the vaccination program in future. And essentially what I'm going to be doing in the next 10 to 15 minutes is providing a very short update on each of these, really the first 3 objectives. So first of all, we can talk a little bit about HPV vaccination. I guess that's really my passion. And definitely of the three pillars, the pillar where we have made the most progress. So our service delivery model for HPV vaccination is that we deliver it through the integrated school health program. And historically, we have always delivered it with 2 rounds, 6 months apart. So the first round of the campaign in February and March, and the second round in later in the year. And that, of course, was governed by the fact that we were providing 2 doses to each girl, and those 2 doses needed to be 6 months apart. So the HPV vaccination campaign was launched by, in fact, our current Minister of Health in 2014. We have used the bivalent Cervical vaccine, and as I indicated, historically we gave it as a 2-dose schedules, with the 2 doses being given 6 months apart. When we started the HPV vaccination program, we targeted girls in grade 4, aged 9 years and older. But in 2020, we moved to grade 5 girls. The reason being that we found that there was a significant proportion of grade 5 girls who had not yet turned 9 years. And although we tried to go back the following year to vaccinate them, it was thought to be much better just to move the vaccination program to target grade 5s, so that the vast majority of them would be 9 years and older. And in this period, 2014 to 2023, a little bit of variation over the years, but 85 to 89% of targeted girls received at least 1 dose of the vaccine. Just to mention that the HPV vaccination campaign is funded through a conditional grant, and the vaccine cost is approximately 350 Rand per 2-dose vial. So vaccinating almost 500,000 girls each year does constitute a considerable investment. Oh, excuse me. One of the features of the HPV campaign is that, as I indicated, it has been implemented by the Integrated School Health Campaign, the health program, which is a program jointly implemented by Department of Education and Department of Health. And schools and educators have played a key role in social mobilization and provision of information to girls and learners, distribution and collection of consent forms, and assistance with coordination on the day of administration. So the model is that during the campaign, we recruit additional vaccinators. The vaccinators move from school to school. Access is arranged by Department of Basic Education, and as indicated, schools and educators play an important role. So that was really how we did the vaccination program from 2014 until 2023. But things have changed a little bit in 2024. So we completed the first round, as usual, in February and March, with the target being grade 5 girls. But we have transitioned from a 2-dose to a 1-dose schedule, in line with WHO and NAGI recommendations. So going forward, the second round of the HPV campaign will be used to provide vaccination to girl learners in grade 5 to 7 in private schools, as well as girl learners in grade 5 to 7 in public schools who have missed their doses in previous campaigns. We anticipate that vaccination in private and independent schools will be relatively limited this year, but it is taking place. And we hope that it is a foundation that we can build on in future years, so that we will be offering vaccination to all girls in South Africa, including the relatively small number. It is about 10% of girls in grade 5 who are in private or independent schools. So it will not affect our coverage significantly. But nevertheless, we feel that it's the right thing to do, to be offering the vaccine to all girls who would benefit. One of the unresolved issues is whether a 2-dose schedule is sufficient for girls who are living with HIV. And so we are looking very closely at that data. And if it is found that those girls do need 2 doses, they will receive their first dose during the school campaign, but they will also be offered a second dose as part of their HIV care in our health facilities. I think I can skip through this slide, but it really just shows the main components of the HPV vaccination campaign. Social mobilization and communication has obviously been very critical. We are worried about spillover vaccine hesitancy from the COVID-19 vaccine rollout, and we particularly anticipate more vaccine hesitancy in the public sector, in the private and independent schools, as opposed to public schools. So we have had quite a wide range of materials which have been made available to our health workers and to our colleagues in Department of Education, so as to provide information and advocacy and to encourage parents to have their daughters vaccinated. Just a little bit about HPV vaccination coverage, because there are some wildly incorrect figures that do circulate. So in this slide, you can see the number of girls that have been vaccinated. We have provided just over 7 million doses, of which about 4 million doses were provided in the first round and about 3 million doses in the second round. Now, the vast majority of doses in the first round are first doses, and those in the second round are second doses. 2 things to note. As I indicated, we reach about 85 to 90% of girls in public sector schools. But in line with WHO recommendations, what we monitor is our coverage in girls 10 to 15 years. And as you can see in the figure, we estimate that we have vaccinated approximately 73% of girls 10 to 15 years of age. So a significant achievement, but nevertheless, we're still missing almost 1 in 4 girls. Why are girls not vaccinated? We do reach almost all the schools, public sector schools with grade 5 girls. Because it's done in the schools, we only vaccinate girls who have a signed consent form. So the main reasons for girls not being vaccinated are that they are absent or that they do not have a signed consent form. When they do not have a signed consent form, it's not always apparent whether that is an active choice by their parents for them not to be vaccinated, or it is simply a logistic issue that the family has not returned the consent form. The parents may not be living in the same household as the daughter. So there are some challenges in that regard. We do try and return to schools to vaccinate girls who are absent, but that is not always possible. The other thing just to note in the slide is the figures for 2020. What you can see is that very few girls were vaccinated in 2020. However, just to flag that we were extremely lucky because that coincided with the transition from vaccinating girls who were in grade 4 to vaccinating girls who were in grade 5. So only a small number of girls were vaccinated, grade 5 girls were vaccinated in 2020. That was because there were very few girls who needed to be vaccinated, because the vast majority of them had been vaccinated when they were in grade 4 in 2019. So this has caused, people have looked at these figures and thought that there was a cohort of girls who missed vaccination, but that was in fact not the case. And it was lucky that we were transitioning during that year. Just a few current issues and challenges. We have moved from 2 doses to 1 dose. We are trying to provide catch-up doses. Obviously, there are issues about changing to the quadrivalent vaccine or even the nonavalent vaccine, and we are often asked about vaccination of boys. So currently, our vaccine selection is governed by the National Advisory Group on Immunization. They are looking at these issues, but it is unlikely that we will offer vaccination to boys. There are other vaccines that we feel should be introduced ahead of HPV vaccination for boys, notwithstanding the fact that if we had unlimited resources, we would almost certainly consider vaccination in boys. I think in the interest of time, we can move on. This is the last slide on HPV vaccination, which really just asks the question, what are the success factors? What are the factors that have contributed to the success of the program? And our assessment is that we have had high-level political commitment from our departments, but also from civil society, huge support from school governing bodies and from parents. We have had dedicated funding from National Treasury through the conditional grant. And then probably the biggest success factor has been the close collaboration between the Departments of Health and Basic Education. So that's a little bit about HPV vaccination. We are hopeful that if we offer a catch-up vaccination and vaccination in independent schools, we will get closer to the 90% target, although it will still require a lot more work and a lot of advocacy to ensure that all girls are vaccinated. So if we move on then, and I'm going to just not talk about these in so much detail, but if we look at Objective 2 and Objective 3, what are our screening and treatment approaches? These are outlined in our policy and guidelines. So with regards to screening, we have 2 different targets, if you like. The first being for HIV negative women. We aim to screen women 30 to 59 years of age, and they should be offered screening at 30 years, 40, and 50 years. So sorry, these are HIV negative women. We would like those women to be screened 3 times. And if they have 3 negative HPV tests, then no further tests will be offered. The situation for women living with HIV is very different. We know that these women are at much higher risk of developing cervical cancer. So we would like to target women from 25 years of age. And these women, sorry, this slide has somehow gone a bit awry. But these women should be tested at least every 3 years, and screening should be lifelong. With regard, just a summary of our treatment approach, we need to make sure that we have all treatment interventions available, especially colposcopy, thermal ablation, and LETs. But the really critical issue that we need to address with regards to scaling up or making treatment more available is the need to ensure that we move services much closer to women. So historically, women have been screened at primary health care, but have then needed to be referred to regional or tertiary hospitals in order to receive treatment. So going forward, there's going to be much more of a push to make sure that all district hospitals in South Africa are of which there are approximately 350, are able to provide at least first level treatment for women who are shown to test positive for HPV, or who are have signs of other precancerous disease. So so I think I've touched on this already, but this is our analysis really of the current challenges facing us with regards to screening and treatment. We know that we have a lot of inefficiencies in our current treatment, screening and treatment processes. I've already talked about the fact that our treatment is extremely centralized. It can often be fragmented. And we know that this places an economic burden and financial strain on patients and the health system as a whole. So this is something that has not yet been finalized, but our key turnaround strategy, our interventions that we will be focusing on, is on the 4 pillars that I've outlined. Then I've talked about decentralized treatment at district and community levels, with the first target being to make sure that all district hospitals, as I've mentioned, are equipped to provide first level treatment services. We know that that is going to require significant investment to make sure that these services have the right equipment and are also capacitated with regards to human resources. We want to have effective screening methods for early identification. So that really speaks to the issue of transitioning from liquid-based cytology to HPV testing. And I'll talk a little bit about where we are in that regard. And then, of course, building this into a comprehensive and coordinated system, continuum of care, which starts with screening, moves on to treatment, but also includes social mobilization and surveillance. So just a few words about introduction of HPV screening. This is the stated policy of the department, and all provinces did commit in 2023 to ensuring that at least 2 districts transition to HPV testing during this financial year. Where we are at the moment is that all districts in KwaZulu-Natal have transitioned. That is 11 districts, and some districts in Eastern Cape have started with HPV testing. 1 or 2 provinces have indicated that they are ready. They were disrupted by the problems at NHLS, but currently support visits are taking place to all provinces. Training is being made available to provinces, and we are optimistic that we will achieve the target of making sure that at least 18 districts do transition during this financial year. We are worried that some provinces are being left behind, and that not all so that there will be 18 districts, but not necessarily distributed evenly across the country. I think I can skip through this. This is the default algorithm that is being recommended for the response to a positive HPV test. We know that this is an algorithm that is designed to be implemented at scale on a population level. And unfortunately, it is a 2-step algorithm because we do not currently have sufficient capacity to follow up directly all women who test HPV positive. We are aware that some centers do have that capacity, and there is allowance for provinces, districts, or facilities to use a different treatment algorithm. if they are confident that they have capacity to provide that service. However, this is the algorithm that is being scaled up across the whole of KwaZulu-Natal, and so we will be able to evaluate and track progress going forward. I think I've spoken about this slide. Finally, then just the last word about the mother-daughter campaign. You know, people sometimes ask, why is it called the mother-daughter campaign? So the mother-daughter campaign is primarily the communication side of things, where we really want to try and link HPV vaccination with cervical screening, and to promote understanding that these two interventions go together, to use our HPV vaccination campaign as an opportunity to intensify messaging and to provide additional improved coverage of our cervical screening of women across the country. So that really brings me to the end of the presentation. With regards to Objective 4, that is work in progress. We have been promised by our colleagues in the digital health space that we will have an electronic vaccination system by 2025. And we're very excited about that. And we are also engaging with NICD to make sure that there are linkages with the cancer registry. So that is my last slide. I think I have gone a little bit over time, but I hope I have provided an update of some of the key thinking and activities from our side as the National Department with regards to cervical cancer elimination. Thank you very much, and back to the chair.
Thank you so very much, Dr. Bramford, for such an informative session. And it was clear and concise. We forgive you 100% for going a little bit over. We really needed to have this background in mind. Thank you for that. Colleagues, please remember to post your questions in the Q&A for later discussion. We are going to move on to the second presentation, but Dr. Bramford is still with us. And at the end of the next presentation, we will then take all questions. Next up, to talk to us about HPV genotypes and the etiology and pathogenesis of cervical cancer, we have Dr. Atisha Maharaj, who is a gynae oncologist at Inkosi Albert Luthuli Academic Hospital in KZN, and she's also an honorary lecturer at the Nelson R. Mandela School of Medicine. Dr. Maharaj is a member of CMSA and served as an examiner and co-convener for Gynae Oncology Certificate exam since 2022. Dr. Maharaj is also a member of the HPV and Cervical Cancer National Advisory Committees. We are privileged to have you amongst us, Dr. Maharaj. You are more than welcome. And I hand over to you at this point. Thank you, Dr. Maharaj.
Thank you, Azi, for the introduction. And I'd like to thank everyone involved again for the invitation to speak tonight. So for my talk, I'm going to be focusing on the human papillomavirus, which in turn is sometimes my best friend or my worst enemy, depending on my feeling to work for the day. It keeps me on my toes, but also it breaks my heart to see the amount of patients that are afflicted by the cancers that are caused by this virus. The human papillomavirus was first linked to the development of cervical cancer by German virologist Harald zur Hansen in 1983, and the discovery of the oncogenicity of the virus was fundamental in the history of medicine, as it made it possible for us to identified the origin of a very frequent tumor, and it paved the way for the development of diagnostic and preventative tools that have been described by the previous speaker. It has been identified as the cause of approximately 5% of all cancers worldwide. It is associated with virtually all squamous cell carcinomas of the cervix, and a significant proportion of anogenital, that is vulvar, vaginal, penile, and anal, as well as oropharyngeal cancers.
Dr. Maharaj,
Yes,
are you on the introduction slide or have you moved to the next one? We
No,
are
I'm
seeing the introduction slide.
I'm still on the introduction slide.
Okay, thank you, just confirming. All right.
I just like to talk, so
Oh, go ahead.
maybe on the same slide for a while.
Okay, sorry for the interruption, Doc. Thank you.
No problem. So HPV infection is also associated with other skin and mucosal lesions, such as warts and benign papillomas. It is also the main cause of pediatric respiratory papillomatosis, which can cause significant respiratory obstruction in those patients who are afflicted by it. This is a world map looking at the prevalence of HPV among women with normal cervical cytology. And while the main focus for eradicating HPV infection is in preventing cervical cancer, it must be acknowledged that HPV does not always cause cytological abnormalities. It shows that the prevalence of HPV infection in women without disease is prevalent in most areas of the world, which is good for them because it means the immune system is preventing development of disease, but they are carriers with the potential to spread the virus to others. 9 out of every 10 sexually active adults at some point will have harbored an HPV infection. The following maps look at the incidence of HPV-related anogenital cancers around the world. The first map looks specifically at cervical cancer alone, and as you can see, sub-Saharan Africa is colored in a darker shade of red. It's almost black, which is in keeping with the knowledge that cervical cancer is higher in lower socioeconomic areas. Areas with the lower incidences, so your pale pink areas, tend to be your higher socioeconomic countries, which have effective measures in place to prevent and control cervical cancer. In the second world map, we look at the incidence of other anogenital cancers, and you can see the incidence rises in those countries with lower cervical cancer incidences, and a fair bit of it is related to disease and sexual behavior, and we'll cover that a bit more towards the end of the presentation. This table is sourced from the International Agency for Research into Cancer that looks specifically at the incidence of HPV-related cancers in Africa. I have highlighted the numbers for Southern Africa, and as you can see, there's a significant number of cervical cancer cases and an unacceptably high mortality of around 50%. This is also true for vulval cancers, which although has a significant lower incidence than rates associated with cervical cancer, it also carries a 50% mortality rate, which is very concerning for a cancer that is outside the body and can be easily detected. Of the countries clustered in the Southern African group, South Africa has by far the most number of cases of cervical cancer, around 10,000 out of the 12,333, and this could be attributed to the screening programs that are in place. However, the impacts of improved screening on mortality rate is not evident, with far too many patients being diagnosed at an advanced stage and subsequently dying from this. The majority of HPV infections do not cause symptoms or disease and are cleared within 12 to 24 months. Only a small fraction of those infections that persist or progress to a pre-neoplastic lesion result in cancer. Each infection stage and behavior may be influenced by environmental, host, and viral factors, the knowledge of which is essential to understand the natural history of the HPV infection, as well as to allow us to develop new tools for improving the management of HPV-positive lesions and the prevention, early detection, and treatment of these cancers. HPV belongs to the family Papillomaviridae, and it's a small double-stranded DNA virus. They are non-enveloped, they are highly tissue-specific, and infect both cutaneous and mucosal epithelium. On the basis of the genomic sequencing of L1, which is the gene encoding the principal caspid protein of the virus, over 200 HPV viral types have been identified and are characterized mainly by the potential to induce cancer and are broadly divided into high-risk and low-risk subtypes. There are currently 12 HPV types that are defined as high-risk and can cause cancer in humans, and these are type 16, 18, 31, 33, etc. Type 16 is known to be the most oncogenic of these types. This table further divides the HPV by oncogenic risk and the associated diseases. The high-risk oncogenic types, which is your alpha genus of the subtypes, are known to cause cancer, while the low-risk types, or your beta and gamma papillomavirus infections, the most common of which are HPV 6 and 11, are known to cause benign warts in specific patients. Generally, these subtypes cause self-limiting infections, but in the background of patients who have immunosuppressed states, such as those with HIV, patients who have had organ transplants and are taking immunosuppressive medication, those with uncontrolled diabetes, etc., they have been shown to exhibit benign disease. There are other groups specifically where you have your probable carcinogenic and possibly carcinogenic subtypes that are really found in large groups or large series of cancers, but they are known to be associated with other risk factors and therefore cause cancer. An example of this is the possible carcinogenic subtypes of HPV 5 and 8, which is known to cause squamous cell carcinoma in the skin of patients that are affected by a dermatological condition of epidermodysplasia verruciformis. It does not cause squamous cell carcinoma of skin in patients who do not have this dermatological condition, and therefore the actual oncogenicity remains to be clarified. So most data on HPV is related to cervical infection and disease, and it's quite obvious because you can see the large numbers that are diagnosed annually. However, infection at all other sites follow the same pattern. So information that's taken from the cervix can be extrapolated to include the other sites of the vulva, vagina, anus, penis, and head and neck. HPV 16 remains the most common form of oncogenic HPV type found across all HPV-related cancers, regardless of absolute number. So we're going to move these various subdivisions in my talk just to break things up so it doesn't seem like one thing is flowing to the other. And the first part of it, we're just going to briefly describe the HPV genome. The viral genome remains the same across all 200 subtypes, with the ability of each subtype to cause cancer depending on the function of its proteins and timing of activation. It's approximately 8 kilobytes in size and is structured into 3 main regions. The early region, which is designated E, encodes genes that are necessary for the viral cycle, with an important role to play in cell transformation. E1 and E2 are responsible for viral replication and transcription. E4 facilitates viron release. E5 is important for alteration of signaling pathways and major histocompatibility downregulation, which prevents immune recognition and response by the host. E6 and E7 proteins are responsible for cell cycle disruption, and all E proteins play a key role in persistent infection and cell transformation. The late region, designated L, encodes L1 and L2 caspid proteins, and these are responsible for viral expression. The upstream regulatory protein, which is a central part of the virus, is referred to as the long control region, as the non-coding region containing the replication origin and transcription factor binding sites that contribute to regulate DNA replication. Also, please note, I have not left out E3 intentionally. It is simply does not exist within the virus. I do not know why. Maybe it had an aversion to it. Going on to transmission of the virus, it's pretty well known that skin-to-skin contact can transmit the virus. And the most common way it does this is through sexual contact. And this can happen through any type of sexual contact. It does not have to be penetrative sex for transmission to occur. It can also occur in non-sexual contact. For example, when you have a mother who has HPV, maybe on her hands, or she has little warts and she has a baby and she's changing the baby, this can cause transmission of the virus from the mother onto the babies. And we see a fair number of children with HPV-related vulval and anogenital disease simply from transmission from an infected mother while they were babies. You can also have vertical transmission of the virus, which can occur during a delivery, where you have passage of the baby through an infected birth canal. And this usually results in infection of the airway. And this gives rise to pediatric papillomatosis, which we see a fair number of it here at Lutuli, with our ENTs really struggling to get control of those diseases. In fact, it's not only the children that are afflicted by this, but it often persists into adulthood as well. Risk factors that increase the risk for transmission. You have HIV-related immunodeficiency, which has complex effects on female genital HPV, which includes increased risk of infection with multiple types of HPV, persistence of virus, reactivation from latency, and an increased risk to develop pre-invasive and invasive disease. Other causes of immune suppression, as mentioned previously, also increase persistence and prevalence, and multiple ulcerative and inflammatory genital conditions, genital infections, such as Neisseria, gonorrhea, Trichomonas vaginalis, syphilis, herpes, and chlamydia, also increase your risk of HIV acquisition, which in turn increases the risk of HPV acquisition and persistence. Additional risk factors for transmission include age of sexual initiation, the number of sexual partners, as well as concomitant sexual partners. This is schematic, a diagram, looking at the crude age-specific HPV prevalence in women with normal cervical cytology. Again, these are women who have HPV but do not have a disease. And you can see a high incidence of infection in those patients under the age of 25, with a sustained 5 to 10% decrease over the age groups as relation stabilizes, and patients tend to have one partner and become more faithful up until your menopause, and then you reach age 64, where you see a sharp upswing in infections. And this is mostly because you have a change in immune system, immune status. So these patients can acquire HIV at a late stage. They may develop diabetes, but also the longer you live, the more likely you are to accumulate DNA aberrations and develop cancer from that point of view. This diagram also looks at the determinants of HPV prevalence by age group. You can see if you look at axis A, the younger you are, that's your age, you have a sharper upswing towards getting HPV. And this is also dependent on the number of partners that you have. A new infection is detected soon after the first sexual contact with an infected partner, and most of them will be detectable within 1 year of exposure. As a result of this high transmission, HPV infections are very common in young women, which depicting the peak in prevalence observed in the figure, which is around 25 years of age. This pattern, as seen in the graph, is consistent in populations all over the world and confirms the sexual transmission as the main mode of transmission. Globally, the prevalence of genital HPV infection in men is higher than women, but persistence is less likely. In general, genital HPV prevalence in men is generally well correlated with women prevalence, but tend to vary less by age group, remaining constant or slightly decreasing with age. Moreover, the detection of HPV infection in men is influenced by sampling technique and also by presentation, because getting men to go to the clinic for something as simple as a STI is extremely difficult. And we find it very challenging even with our patients we have diagnosed with HPV and having genital disease to get their partners to present for treatment. It is extremely hard. And that may represent a bias in our numbers that we have gathered. However, males generally have less persistent infections and therefore have less HPV-associated cancers when you look at absolute numbers compared to females. So moving on, HPV is a virus. It should induce an infection. So the question is, why doesn't our immune system clear the infection in time to prevent disease? And that is because HPV is an extremely, extremely clever little virus, especially the high-risk genotypes that have developed several mechanisms over the years to avoid host immune response, which is important for viral persistence and progression to HPV-associated neoplastic disease. One of its first strategies to avoid detection is to maintain a very low profile. The HPV life cycle is exclusively intraepithelial and non-lytic. If you compare, for example, to a bacterial pneumonia, where you have bacteria that infects the cells, parenchymal cells of the lung, it does eventually end up killing the cell. And the cytokines released by the cell death is what alerts the immune system to the fact that there is an invader. HPV doesn't do that. It does not kill the cell. It changes it slowly, so to suit its own needs, and therefore prevents the associated pro-inflammatory signal that is associated with all infections. As a result, the recruitment of antigen-presenting cells and the release of cytokines are absent or very low after initial HPV infection. So you can see in this slide, you have your basal layer, and then you have your subsequent layers of epithelium. And your basal layer is where the virus starts. And it stays there, changing the cells, as you can see slowly, as it goes from pink to purple. And then it affects cells adjacent to it. And it does this very, very slowly, which is why disease takes time to develop And disease is really only evident a few years after initial infection. It builds up through the layers until it reaches the surface epithelium, and it is an extremely patient virus. That is why we need an effective immune response, and that is why the vaccines were developed, because that allows presentation of viral-like particles to lymph nodes in the region where the injection is administered, and allows a more rapid response as compared to natural infection. So what happens when you do get an immune response? So if you look at this diagram, you can see your infected cell in the basal layer. It's differentiating through the layers till it reaches the surface, and you have active infection that has alerted the immune system. It sends T cells to the infected area, and you start getting regression of your lesion until you get latency back in the basal layer. This cell may remain here undetected for years, but it can become reactivated from latency with changes in immune status, as we previously described. And it starts all over again, differentiating through the layers until it reaches the top. Disease persistence corresponds with the failure of the immune system to properly detect infection. And as we said, it's because of the low numbers of antigen-specific T cells. In an immune-tolerant environment, disease persistence is facilitated and deregulated viral gene expression can go unchecked. For the high-risk alpha HPV types, the loss of control by key HPV genes, particularly the E6 and E7 genes that regulate cell cycle entry, proliferation, and differentiation, leads to dramatic change in lesion phenotype, which lead to development of cervical intraepithelial neoplasia, grade 2 and 3, and subsequently to invasive disease. So just as a summary of what I've gone through, that the majority of HPV persists quietly within the human host as they delicately modify the epithelial homeostasis and cell function upon infection, and that sets them up to allow persistent chronic infection without clearance because it is continuously evading the immune system. The life cycle starts at the basal layer through microtraumas that compromise the epithelial barrier. It stays at a low copy number, and then it differentiates upwards. This is a schematic diagram of what happens. Sorry, I'm going too far. Okay, so you have your basal layer in green here with your initial infection, and as you can see, it slowly works its way up to your suprabasal layer, where you have genomic maintenance and cell proliferation by your E1, 2, 5, 6, and 7 genes. As it continues up, you can see there are more viral parts that are released with genomic amplification, and this goes on to your granular layer where you have virus reassembly by the proteins L1 and L2, and then you have viral release in your conified or stratified epithelial layer. So if you are looking at, just to put some clinical impact of this, when you have basal infection here and you do a cytology smear, the result will come back as HPV changes but no dysplasia. When you have reached the suprabasal layer, your cytology will show you a low-grade lesion or CIN1. Up to your granular layer, it's CIN2, and up to your stratified epithelium, you have CIN3 or full thickness dysplasia. So it is at this point when it's at CIN3 that the immune response is most robust. But by then, it is too late because you have infected cells all the way up. So why does HPV infect the basal cells? That's because basal cells are shown to have stem-like features because they're still able to proliferate despite having damage. But this is more common to HPV high-risk subtypes, and that is why they are responsible for most of the disease. The epithelial transition zones, such as the endo and ectocervix and anorectal junctions, are regions that are more susceptible to carcinogenesis by high-risk HPV types. They are more prone to activate cell proliferation in basal and differentiated layers, promoting the transition from productive infection to an infection which is unable to complete the viral life cycle, but is able to activate several pathways essential for the epithelial transformation required to develop malignancy. So why are the high-risk types so oncogenic, and particularly HPV-16? And the reason for this lies mainly in the activity of the E6 and E7 oncoproteins. And while they are present in both high and low-risk subtypes, the role in low-risk subtypes is limited to increase the viral fitness and viral production, and it is largely insufficient to trigger the development of pre-neoplastic lesions and cancer. So how does the cancer come about? So like mentioned, E6 and E7 are responsible for malignant change and are more active in the alpha genus, so your 16 and 18 and friends, and they mediate carcinogenesis by the inhibition of two key tumor suppressor genes. Just going to go through them briefly. So the first one is P53. It's a well-known tumor suppressor gene and is commonly referred to as the guardian of the genome, as it is responsible for maintaining DNA integrity. It does this by interrogating cells that enter the cell cycles, and any cell that exhibits DNA damage by any cause is halted. It is further interrogated to find what is the fault, be it by DNA damage, hypoxic injury, chemotherapy, etc. And if it finds that DNA repair is possible, the cell is sent through a repair process and allowed to re-enter the cell cycle. If it is considered to be irreparable, it induces apoptosis and the cell dies with its abnormality. The second tumor suppressor gene involved is the retinoblastoma protein, which is another tumor suppressor gene that is responsible for regulation of cell growth, and it stops cells from dividing too rapidly or in an uncontrolled way. Remember, the hallmark of any cancer is uncontrolled growth, and therefore the retinoblastoma protein is important in preventing this, not only for cervical cancer, but for a majority of cancers. E6 and E7 early proteins conduct a main role in the carcinogenic process through the inhibition of P53 and the retinoblastoma tumor suppressors. E6 specifically targets P53 by activation of telomerase activity, as well as deregulation of pathways involved in immune system response, epithelial differentiation, cell proliferation, and survival signaling. E7 specifically targets the retinoblastoma protein and results in cell cycle deregulation and proliferation, which enhances genomic instability and allows for the promotion of the accumulation of chromosomal abnormalities. The deregulation of the cell cycle, the activation of telomerase activity, and genomic instability create a favorable environment for epithelial cell transformation. Excuse me. This diagram just summarizes the carcinogenic process initiated with E6 and E7. However, Activation of E6 and E7 alone cannot be held responsible for carcinogenesis. It also needs to be complemented by the accumulation of additional alterations in the host gene to lead to the invasive cancer phenotype. Viral genetic variation beyond the HPV genotype could explain in part differences in clearance, persistence, and the risk to develop cancer among infections positive for the same type of HPV. There's also a role for epigenetic alterations, such as promoter methylation in certain cases, or loss of DNA proteins. Environmental and exogenous factors identified as modifiers of the natural history of HPV infections are also to blame. Other risk factors that are associated include long-term smoking, multiparity, long-term hormonal contraception use, co-infection with sexually transmitted infections, and of course, we cannot forget HIV, which is an associated major contributor due to the related immunosuppression. So among the many viruses that have evolved with the human hosts, most papillomavirus infections are either asymptomatic or inapparent, or lead only to the appearance of benign self-limiting lesions that are not life-threatening. This balanced relationship ensures survival of the infected host without compromising evolutionary fitness of the virus itself, and this allows for continued virus transmission and persistence within the population as a whole. So that is why we look at population-based strategies to eliminate HPV, because by eliminating the virus itself, that is the only way we can aim to eliminate HPV-related cancers. So I'm going to leave virology and theory behind for the last part of my talk, and we're going to discuss HPV field effect. I really wish it was about a field of roses or tulips, but it's not as fun. The term field effect was originally described by Slaughter in 1953. It postulates that a single stem cell undergoes a genetic transformation, which then results in daughter cells acquiring similar genetic alterations. As long as the benign fields contain the genetic aberration, these are bound to progress to either pre-malignant or malignant lesions at some later stage. The transformed cells tend to involve contiguous organs. The term field effect of HPV is not often used in literature, but it is the same as when you have a genetic aberration as when you have infected cells. This field effect of HPV refers to the involvement of different sites, so your cervix, vagina, vulva, and anus, with multifocal disease. These lesions may occur synchronously or metasynchronously, and there's very little research published on women who have multifocal disease. This may be due to underreporting, or rather a focus on cervical disease only with less examination and note taken of the other genital areas during colposcopic examination, and therefore these are missed. The pathogenesis driving the field effect is unclear, but follows three possible theories. The first is the theory of monoclonal spreading, which refers to a single clonal cell that undergoes genetic changes. These changes vary from benign to pre-malignant and malignant in the same organ or tissue in close proximity. Secondly, it is the theory of persistence of high-risk HPV infection. And the last theory is that you may have synchronous infections with several different types of HPV, which result in multiple different lesions. Immunocompromised women, especially HIV-positive women, are more likely to present with a field effect of HPV-associated disease. This is due to their impaired immunity and inability to clear the infection with subsequent persistence of HPV. And you have co-infections with multiple types of HPV. As a consequence, there's an increased incidence of lower genital tract malignancies in these immunocompromised women. Studies where HIV and cancer registries have been linked, it does reveal a 22-fold increase in cervical cancer in HIV-positive women when compared to HIV-negative counterparts. There's also an increase in incidence of cancer of the vulva, vagina, and anus. So what about screening for HPV disease? Screening for the cervix is well established with cytology and HPV DNA testing. Screening for HPV of the vagina, routine screening is not performed, but examination of such should be included in a complete colposcopic examination. Screening for the vulva is not routinely performed, but inspection of the area in women who are at high risk of developing the disease should be sufficient. HPV in the perianal area, the role of screening has not been established, but we do know that the use of perianal cytology misses approximately 45% of precancerous lesions. So coming back to my world maps, this is the one I showed at the beginning of the presentation. And to show an example of this, I'm going to pick on Australia. And if you look at the cervical cancer map, Australia is light pink, which shows a very low incidence of cervical cancer because it has a very effective screening program. However, you do have an increase in incidence of other anogenital cancers in the same region simply because they do not have effective screening programs. Not only them, we don't have effective screening programs for the other anogenital cancers. So it does show if you have a screening program that's effective, that's implemented correctly, you can decrease your incidence of cancer. So how do we manage these patients with field effect HPV? Almost all of them require a combination of excisional and ablative treatments. Defunctioning stomas may be indicated in those with extensive multifocal perineal and perianal disease. Many patients require more than one procedure, and a lot of time we have to involve multiple specialties. These patients require long follow-up post-treatment as the areas remain susceptible to dysplasia and malignant transformation. I'm not sure it was because I was giving this talk, but today in my clinic, I saw a patient that I first treated in 2018 for vulvar pre-malignant lesion, which was HPV related. It was excised, she was fine, but she did have CIN, so cervical intraepithelial neoplasia, which was persistent. She had a hysterectomy done for that at her referral hospital. And she was subsequently referred back to me today because she has now been diagnosed with vaginal intraepithelial neoplasia as well as anal intraepithelial neoplasia. So she has been referred back for treatment. And she, unfortunately, is the perfect example of what we encounter with patients with HPV field effect. But the promising part of the HPV vaccination is that studies have shown that the vaccination at an early stage before sexual debut and exposure have been shown to decrease HPV infection and the subsequent morbidity. So finally, in conclusion, it's almost an appeal from my part, because I can tell you at our clinics at Albert de Tully annually, we treat approximately 750 cervical cancer cases. Only 5% are operable. We treat with a combination of therapies approximately 100 patients with HPV-related vulval cancers. We use excisional and ablative therapies to treat HPV-related vulval and vaginal neoplastic disease for close to 180 patients We're looking at 1,000 patients of HPV-related disease that we treat in our center alone every year. And that can be extrapolated to our other centers in KZN, centered in PMB, and up in Queen Nandi Hospital. The important part is this affects women that are in the prime of their lives. They are mothers, they are homemakers, they're caregivers, they are breadwinners. And yet they are afflicted with a disease that is debilitating, but wholly preventable. And this is why we need to promote and encourage preventative strategies, such as vaccination and screening, as this is the only way to lift the burden from our patients. Thank you.
Thank you very much, Dr. Maharaj, for that wonderful talk. Very technical at points, but thank you. We really appreciate how you broke it down so that we can follow. Before we take questions, Dr. Maharaj and Dr. Bramford, could you please show us your faces? Yebo. Hi. Let me greet properly in a video also. Hi, Dr. Bramford. Hi, Dr. Maharaj. Thank you very much. We are going to start with the question and answer session. I think if you are comfortable, we can switch off the video or switch them on. It's your prerogative. But let's start first with the question that is more from a programmatic perspective. Dr. Bramford, there is a question that has been asked, and I will read verbatim. Is there provision made to vaccinate boys as well?
So I think I did touch on this in my presentation. And the answer is that we do not anticipate that in South Africa we will start vaccinating boys in the foreseeable future. We know that there are benefits for boys, but I think this one of the slides that Dr. Maharaj showed the burden of disease. I don't know if you remember the slide. And the cervical cancer, the bar was very, very long. Mm-hmm. And then the bar for the other HPV-related cancers, which are the ones that also affect boys or men, the burden of disease is much lower. So I think, you know, we have a pot of money and we have to make decisions about what we fund and what we don't fund. And in fact, there are some other vaccines that probably overall have more benefits than the benefits that we would see from providing HPV to boys. So our commitment is to try and reach every girl because they have a cervix, and we need to try and prevent cervical cancer. I obviously, you know, if resources were not an issue, we are very well aware that there are benefits for boys, and we might want to offer it. Obviously, males also play a role in transmission. But my understanding is the advice from WHO is that actually the best place to put your resources is a well-functioning program targeting girls. And if you do that, you will protect them and also develop some herd immunity. Thanks very much.
Thank you very much, Dr. Bramford. I see Dr. Maharaj's hand is up. Yes, please go ahead, Dr. Maharaj.
Yes, thank you. I just wanted to add to that. I was also going to mention the WHO recommendations, and their primary target group is the girls between ages 9 and 14. And they recommend vaccination of boys if your coverage is under 50%, or if you have enough resources as to not compromise your primary group. So until we reach that point, I don't think we are going to include boys. And like Dr. Leslie said, probably the next I don't know, 4 or 5 years before we get to boys.
Thank you very much, Dr. Maharaj, for that, and Dr. Leslie. There is another, I'll take the next vaccine-related question while we are here. The question is, shouldn't we extend HPV vaccination to beyond 45 years? Excellent presentation, by the way. I'm reading exactly as it is. Shouldn't we extend the vaccination to beyond 45 years? I don't know if maybe he meant beyond 15 years, I assume, but maybe let's just deal with why are we stopping at 15 years at this point?
So can I start, and then perhaps Dr. Maharaj would like to add? So
yes, absolutely.
So our understanding is that it's for a number of reasons. First of all, we want to start as early as possible for 2 reasons. First of all, we want to start before girls are sexually active. And then we want to start as early as possible because the immune response is best in young girls. We don't vaccinate girls younger than 9 years because the vaccines are only registered from 9 years. But there are trials that are ongoing looking at vaccinating young girls. And so perhaps eventually we might provide HPV as part of our childhood schedule. There's no reason why we shouldn't, but at the moment we have no experience and no data on safety or efficacy. So that's why we start from 9. We go up to 15, I guess, because we know that after 15, then many girls are becoming sexually active. That's one reason, and we want to vaccinate them before they become sexually active. Also, from 15, as we don't have data, so some of the data suggests that we still need to use 3 doses. So that's the answer from a programmatic point of view. Vaccinating older people, in our understanding, is less effective, A, because they're likely to have been infected with HPV already, and because the vaccine is less efficacious. Thanks.
Thank you very much, Dr. Bramford. Dr. Maharaj, anything to add?
Yes, so there is evidence for catch-up vaccination up to age 45. According to, well, it is in the WHO recommendations. You don't need to have an HPV test done. It's not currently offered as part of our vaccination program because, as Dr. Leslie has mentioned, it is aimed towards the most vulnerable group. It is available privately where you can get a script and buy out your own vaccines and take them, but it's only recommended up to age 45 because that's where the research stops. The vaccine producers, as I understand it, have ongoing trials looking at older age groups, but right now the evidence only stops at age 45, which is why the recommendation stops at age 45.
Thank you very much, Dr. Maharaj. And that's a very important point to make because in our audience, we have colleagues who are working in the public sector, but there are also colleagues in the private sector. So thank you so much for raising that, because in the private sector, if one can afford, then they can access the vaccine privately. And even for the boys, if one is passionate about it and they want to vaccinate the boys, yes, privately, you can access the vaccine. Thank you, Dr. Maharaj. I have one before we move on to the next set of questions. And for both of our speakers, colleagues, is there an indication that perhaps in the future, we might need to do boosters? Is this one vaccine, one dose enough for the rest of the life of the girl? Or will we need to boost at any point?
So again, let me start from a program perspective. So, I mean, our understanding is that this is actually a very effective vaccine, particularly if we can give it to young girls. So remember, we started with 3 doses, and we've come down to 2 doses and now 1 dose. And there is good evidence for the period for as long as recipients have been followed up that they are protected. So I guess we don't know in the future, and that is something that would need to be evaluated. But my understanding is that there is no indication that this is anything but a very effective vaccine. So we're not expecting to need boosters. I think one area where there perhaps is still some uncertainty is in individuals who are immunocompromised. And so particularly in South Africa, young girls or women living with HIV. So there is not yet data to show that 1 dose is sufficient in those cases. There is no, as I understand, there's no data to show that 1 dose is not sufficient, but it's a data-free zone. So the default is, we think that we will probably need to provide 2 doses to women living with HIV, but that may change with the future. So as I indicated, our approach would be to vaccinate girls, get their first dose as part of the school program, because of course in that program, we don't know who's living with HIV and who isn't. And then we would look to offer the second dose as part of HIV care. We're not rushing to do that because we're waiting for some of the data, but we are very aware that we may need to do that in the future. Thanks.
Thank you very much, Dr. Bramford. I'm going to move to the next set of questions. And the first one says, the colleague is asking, my concern is that there are numerous cases where patients between the ages of 17 to 25 are presenting with high-grade lesions. What happens if we are only starting to test at the age of 30 years? So let me give Dr. Maharaj an opportunity maybe to explain to us where that comes from. But if even Dr. Bramford, it's okay, whoever wants to go first. The concern is that there are numerous cases of high-grade lesions in patients between the ages of 17 to 25, yet we start to test at age 30.
Okay, so I will start with that one. So the reasons that we chose the ages that are in the guidelines is that we do know, yes, they do have HPV infections and you have high-risk HPV, but because of their youth, they tend to clear those infections more effectively than those in the age over 30. We go earlier in your immunocompromised because obviously the immune system starts to fail a little bit earlier. So if we test in those age groups, and there are countries that do test, the USA example, they start their screening at age 21, you find there's a high level of interventions for these patients with very little benefits, because if given enough time, they will clear the infections on their own. Also remember that treatment of these infections usually involve either ablative or excisional therapy of the cervix, which can affect their fertility potential later on. So we want to avoid a point where we are going to be intervening unnecessarily in a patient who will clear the infection themselves and not develop actual disease. If you may remember in my talk, there were diagrams where you had patients who have high-risk HPV, but actually did not have any dysplastic or cytological abnormalities, and therefore did not need further treatment. It's a very delicate line to walk. So those patients who you do find have high-risk HPV at a younger age, they need to be followed up closely, and the decision to intervene therapeutically needs to be carefully monitored. Thank you.
Okay. Thank you very much, Dr. Maharaj. Dr. Bramford, anything to add, or can I move to the next?
Please move to the next one. Thanks very much.
Okay, thank you. The next question also relates to the age of testing. And the question is, given the fact that the HPV prevalence shoots up after 50 years in our settings, shouldn't we offer HPV testing more frequently after this age band? What are the pros and cons? Dr. Maharaj, I remember that graph you showed us of HPV-related prevalence as you progress through the ages. So, and I remember you were saying we do stop HPV testing, particularly in the actually in the HPV negative. We stop after 3 consecutive negative HPV tests. So I'm thinking of that when I read that question, but the question is clearly looking at, given that the prevalence shoots up after 50 years, shouldn't we offer HPV testing more frequently after this age band?
So not necessarily. So that increase in HPV infections, that's a disease that is picked up in that stage, is usually a reactivation from latency. So which means they would have picked up that infection earlier on in their lives, and if they've had testing, that would have been picked up. The exit from screening is on the provision that you've had 3 normal screening tests. So whether they be cytology or DNA, you have to have had 3 normal tests before you're allowed to exit screening if you are HIV negative. HIV positive patients never exit screening regardless of how old they get. The second part of that is that, remember, this is a guideline only, and it is tailored according to your resources. So in the public sector, we generally don't have the resources to keep continually testing patients. And we have to have a stop point to allow for resources to be directed to where they can be more effective. In higher resource settings, your testing can be done more frequently, and it can be done lifelong, even in your HIV negative woman, provided your resources allow for it. Thank you.
Right. Thank you very much, Dr. Maharaj. I am moving on to another question related to screening. Must we do the pap smear on young women? Let me see if I get it correctly. Must we do the pap smear on young women before the actual age, because they follow their age groups who are on chronic medication and must be screened annually? I don't know, Dr. Maharaj, if you understand, but this, I've read the question exactly as it's stated. Must we do the pap smear on young women before the actual age, because they follow their age groups who are on chronic medication? I'm going to skip the colleague, Dr. Maharaj, if you can get it, you get the question, please do respond, but it's missing me.
So if I don't understand it completely, but there is provision for testing outside the screening guideline, because screening is specifically aimed at asymptomatic patients. So if you have a person who, say, it has symptoms, so you have a younger patient who falls outside your screening age, who's presenting with a persistent discharge or abnormal bleeding, you're concerned, then yes, you can screen that patient, even if they do not fall within the guideline because they are symptomatic. If you have a patient who is at risk, say they've been abused at an early age, and you are concerned they may have been exposed and now have maybe symptomatic, yes, those patients can be screened. A guideline is not set in stone. It is there for the majority of patients, but they need to be modified depending on the patient that you have.
Thank you, Dr. Maharaj. You got the question spot on. There's a response that the colleague is answered. She's saying, he or she's saying, thank you. All right. There was a hand, I don't know if it's still active. One of our colleagues, Dr. Mandondo, had her hand up. Dr. Mandondo, are you still? Yes, I see her. Carolina, is it possible to unmute? Yes, thank you. Yes, good evening, Dr. Mandondo.
Good evening. Thank you very much for this very great talk. I had two questions. This will talk to both Leslie and Dr. Maharaj. In terms of the HIV positive patient, we were screening them at the start of initiation, before initiation of treatment, irrespective of age, but normally it was around the 20s, they were getting screened. And then they were then screened 3 yearly. Now, in terms of these guidelines, there is a suggestion that we should then screen them only after the age of 25 and maybe screen them 5 yearly. So to Leslie, the HAST guidelines do not say that, and not everyone has transitioned. How would we then communicate in a setting where some districts are still doing HPVs and others are not? Should we not, and then also the indicators on DHIS are different. They're still saying HIV positive 20 years and above. How will the data collection, will this not impact the data collection? Or should we not rather do a circular, consult with HAST and make sure that maybe once all the districts have transitioned, then we communicate this? But I would like to hear from Dr. Maharaj as well as a clinician. Are they comfortable with this, considering we've got all these children that were born HIV and the early age of sexual intercourse in our setting? Is this 25, to begin screening in the HIV population really appropriate? And then when do you decide if you're going to do 3 years or 5 yearly? Thank you.
Thank you for the question, Dr. Mandondo. I'll hand over to Dr. Bramford.
Yeah, so thank you. I mean, I think from an implementation point of view, we always struggle with guidelines not being aligned. We do obviously try and align them as much as possible, and it is going to be particularly difficult in the next year or so, when some provinces have transitioned to HPV testing, and some within a province, some districts may have transitioned earlier. So I don't think we have all of those answers, but I think it's a good thing to flag, and it's definitely something that we should give consideration to. I mean, I think we mustn't lose sight of the biggest problem is that women are not being screened. So our best estimate of screening coverage in the public sector is around about 35%, which is really extremely low. So I do think that our first priority is really to increase screening coverage. Obviously, we need to complete the transition to HPV testing, making sure that we are actively following up every woman who does test positive. For me, those are kind of the headline issues. And it is very important that we sort out the misalignments, etc. But maybe that's something that we can take outside of this meeting. Thanks very much.
Thank you very much, Dr. Bramford. Dr. Maharaj, your colleague is asking, are we comfortable as clinicians about this, the age of starting screening being 25 in this population of HIV positive girls and women?
So there's lots of different opinions and arguments that, yes, we should stick to the initial, when you're diagnosed and you're sexually active. I mean, if you're born with HIV, as soon as you become sexually active, you should be tested. And then there are those that also point out that they will clear infection and we may be intervening too early. So the other side to that is that if we are going to increase the age by starting screening at age 25, as opposed to time of diagnosis, a suggestion was put forth that maybe you should have sort of like a screening questionnaire. See, is this patient symptomatic? Is this patient at risk? And should we screen the patient prior to the suggested age of onset? But that is still under discussion. When we look at the intervals between testing, yes, for cytology, initially it was every year for HIV positive, and we went to 3 years because it showed there was no difference in your diagnosis of pre-malignant diseases. So there's no difference in your sensitivity of the cytology test between 1 and 3 years. The reason it has gone to 5 years now between testing with HPV DNA is that it has a higher sensitivity and better specificity to exclude disease than cytology alone, and therefore it allows you to extend your screening periods. And that is one of the reasons you're aiming to transition to HPV DNA, to decrease the cost of the screening program, because you're going to screen less frequently with a more reliable screening test. However, I do agree with Dr. Bramford in that it really doesn't matter which screen you use, whether you use cytological screen or virology screening, as long as we improve our screening uptake. But it is going to be a while before we can collate all provinces to follow a single screening protocol. That's why the HPV DNA one hasn't been published widely, because it's not widely available as yet.
Thank you very much, Dr. Maharaj. I also have a question. In this particular age group that Dr. Mandondo is referring to, is there no role for the use of cytology still, like we used to before this transitioning to HPV era? Would there be no role? Because now we understand the limitation of HPV screening, that we could be picking up infection that the woman can still has a high chance of clearing. But couldn't we use cytology like in the past?
Yes, we could. And this is actually one of the points of discussion that was being had at the meetings, that we should maybe still consider cytological use under the age of 25 for this group of patients. So there's a lot up in the air because of the prevalence of HIV and HPV in our communities before we come up with a final document that says, well, this is what we are going to go with. But yes, I take your point, and I agree it is a potential strategy to use cytology screening before the age of 25.
Thank you very much, Dr. Maharaj. There's a question about vaccines. Colleagues, let me just say next week we have a full blast vaccination talk. But let's talk, because now we have Dr. Bramford with us as well. And next week we might not have her with us. But please don't miss next week's webinar. The question is, any thoughts on therapeutic use of the vaccines in the proven presence of HPV-related pathology in both HIV positive and negative individuals, both male and female. Therapeutic vaccine use in the presence of HPV-related pathology. Any thoughts? I'll start. Okay, Dr. Bramford's mic is on.
I unmuted, but only to say that's definitely outside of my knowledge or experience. We use vaccines for prevention, and they're very good. And I'm afraid I know nothing about using them therapeutically.
Thank you, Leslie. Dr. Maharaj?
So from my side, yes, there's been a lot of talk about using it as a therapeutic option, but there's no evidence to support that it is effective, especially for genital disease. There have been studies regarding respiratory papillomatosis in the pediatric population, where it has shown some promise. And it's ongoing from there, looking at use. And I think that's a very important group that if we're going to use therapeutic vaccines, it should be tailored towards that group. But there's nothing sufficient enough in the anogenital groups to say that, yes, it is feasible and effective. So my answer is no.
Okay, thank you very much, colleagues. If you have more questions on the vaccines, please go and think about them, write them down. We will be back next week going deeper on the vaccines. But in the meantime, let's deal with what we have. Someone is asking, is there any superiority between the vaccines available in private? Is any of the vaccines superior to the other? And I'm assuming the colleague is probably asking about the quadrivalent versus the bivalent versus the nanovalent.
Okay, so I think obviously the 9-valent has viral-like particles to induce responses against 9 different types of HPV, so it should be considered more effective. Quadrivalent covers 6 and 11, which is important for your benign diseases, which, yes, can be considered superior. But in the terms of prevention of cervical cancer, and indeed most HPV-related cancers, type 16 and 18 are the most prevalent HPV subtypes, and that is what you need protection against. There's also the, but it's unknown to say theory, but you also have cross-protection against other subtypes, because all of these high-risk subtypes belong to one genus, and that's your alpha genus. So there will be cross-protection against other subtypes, although maybe not as effective as against 16 and 18. So I often get asked this question by groups. So when I'm talking to medical students, etc., and I encourage them to get the vaccine, and they ask me which one, I tell them, which one can you afford? Because even though you're in private, you may not be able to afford the 9-valent, but you can afford the bivalent vaccine. Take the bivalent because it will protect you against the subtypes that will most commonly cause the cancer. So in terms of prevention, 16 and 18, there isn't any. And the other subtypes are only found in cancers for women and only account for up to 12% of the cancers. So there's no real clear.
Thank you, Dr. Bramford. Any additions?
So I don't think so. I think certainly when we procure vaccines, we consider the vaccines to be equivalent in terms of efficacy, and so then price does become a very important deal breaker in which vaccine we buy, because we do need to be cognizant of the fact that we are in a resource constrained environment, and we do need to provide services that are effective, but that are affordable at the same time. Thanks.
Thank you very much, colleagues. I think our closing question or point of discussion will be based on this comment here. Shouldn't we consider contextualizing vaccination approaches, considering that some African cultures subscribe to polygamy? South Africa should consider vaccinating boys sooner than later, particularly in areas like KZN. Shouldn't we contextualize our approach, considering that we are in a context where polygamy is practiced? Yes, Dr. Bramford.
Yeah, so I think we should really take that as a comment, and we can always consider things, but I think we have answered the question around vaccinating boys. We know that there is benefit for them. We know that they do play a role in spreading HPV infection, but currently our emphasis needs to be on girls who, because we are aiming to prevent cervical cancer.
Thank you very much, Dr. Bramford. I'm just going to read some comments just so you know how our audience felt about this presentation. There's a comment that it's very interesting and informative as a clinical nurse midwife. I'm enjoying, thank you. Also appreciation from Carmen. Thank you for a very informative discussion and analysis of the HPV infection. It is a tremendous help in informing MLS students in understanding how the virus works and affects patients and the long-term effects and devastating effects it could have. Thank you very much for those comments, colleagues. And there's a question about, are we going to receive CPD points and certificates of attendance? Colleagues, the CPD points, absolutely. For colleagues who are registered with the Health Professions Council of South Africa, we are going to be submitting on your behalf, so you don't have to worry about a submission. And in terms of the certificate of attendance, colleagues, we have all your details and we will communicate appropriately. But in terms of the CPD points, we definitely will submit to Health Professions Council of South Africa. Before we wrap up, let me check. My colleague Carolina, could you please share the survey for the webinar? Colleagues, please do not log off before you just give us. It's just a few questions, just to say, you know, did you find the webinar useful? And if you have any comments, please add them on there. And with that, colleagues, I just want to say thank you to our wonderful speakers. Dr. Bramford, it's always wonderful to hear from the horse's mouth, because, you know, we always want to know what is the National Department of Health up to. So thank you so much for gracing us. We had really a good opportunity to understand what is the strategy and what are the key interventions that are being planned for the country. It truly was a treat to have you. And to Dr. Maharaj, I've never had the life cycle of HPV. I know the life cycle of HIV very well, but I really have never been to a presentation where we were taken blow by blow through the life cycle. I have personally learned a lot, and I know that the colleagues also learned so much. It's a very technical aspect, but the way you broke it down, thank you so much, Dr. Maharaj. I can never forget. At some point, we are going to be talking about the tests that are available for HPV testing. And I've been hearing a lot about point-of-care tests for E6 and E7 protein. Now it's beginning to make sense. So when we have that conversation 2 weeks from now, it will be easier for us to understand what exactly are we testing and how to interpret the different tests. So Dr. Maharaj, to you too, thank you very much. Colleagues, to all the specialists, GPs, nurses, everyone who took time to be here today, I know, colleagues, it's because your heart is in the right place. All of us do not want to see a woman who is still in her productive age dying of a disease that they shouldn't be dying from. So, colleagues, thank you so much for that. And I know that we are gathering advocates for vaccination. We have heard about what HPV does. That is not just the cervix that we are, the cervical cancer we are preventing. There are other cancers, but the burden of disease is heavily towards cervical cancer. That's why you are hearing so much about cervical cancer. Colleagues, with that, I'm going to stop because I will start preaching now and people will start having to say amen. So colleagues, thank you very much for your attention. We have kept to time. May you have a wonderful evening. Next week, we are going to be back at the same time on this platform. We will be delving into HPV vaccination and also hearing how other service providers are actually involved, or what strategies are they using to increase their uptake of HPV vaccination. So we will be joined by colleagues from the DISCAM group, and they will share with us how, not DISCAM next week. Next week, we will be hearing about vaccine hesitancy from UNICEF. So colleagues, please join us. And as we leave, I will just put up our schedule for the next 4 webinars. And again, thank you very much for joining us, colleagues. Till next time, adios.
Bye-bye. Thank you very much. Have a good evening.
Thank you. Thank you.
Bye.